students. Today we will continue with our chapter Biotechnology Principles and Processes. In my previous videos, if you have gone through, we have already started with the chapter where we have gone through the details of the restriction enzymes or the molecular scissors, the features of the cloning vectors, few examples of cloning vectors, and how the difference between the transformants and the non-transformants is done. Yesterday I have also discussed the various steps involved in the recombinant DNA technology. In just a I have given just a glance. Today we will go through the various steps of recombinant DNA technology in details. The first step is the isolation of the genetic material, which is usually the DNA and RNA in some cases, mostly the viruses. Now, RNA, if it is in case of DNA, that means RNA has to be removed, right? So, RNA is removed by the treatment with ribonuclease, and proteins are removed by the treatment of protease. Then, after several treatments, the purified DNA is precipitated by adding chilled ethanol. If we have I have not given you in practical, ethanol, chilled ethanol always helps to precipitate the DNA. Okay, the bacterial cell or the plant cell or the animal cell is then broken down by the enzymes to release the DNA along with the RNA, proteins, polysaccharides and lipids. Now, the bacterial cell is treated with the enzyme lysozyme. If it is a plant cell, we need to break the cell wall. That's why it is treated with cellulase. And if it is a fungal cell whose cell wall is mostly made up of chitin, so it is treated with chitinase. Next, we will come to the cutting of DNA at specific locations, and this is done using restriction enzymes. Okay, so the purified DNA is first incubated at a particular temperature and then treated with specific restriction enzyme at conditions optimum for the enzyme to act, because enzymes act only at an optimum temperature. Now, next is isolation of the desired DNA fragment. And this is done by using the agarose gel electrophoresis, whose details we have already gone through, and the activity of the restriction enzyme can also be checked in the gel electrophoresis. Now, since the DNA is negatively charged, it moves towards the positive electrode or the anode, and in the process the DNA fragments actually separate out based on their sizes. Now, this desired DNA fragment is then isolated. The next step is the amplification of gene of interest using PCR. Now, what is a PCR? PCR refers to polymerase chain reaction. Polymer, as the name suggests, it is a polymerase. That means it will produce a polymer of the given sample. Now, PCR is actually a reaction in which amplification of specific DNA sequence is carried out in vitro. It is in vitro because it is carried out inside the test tube. Amplification refers to it produces several number of copies. The number of copies we want. Because it is artificial, we can actually produce the number of copies we want. Okay, and this technique was developed by Carey Mullis in 1985, and he also received a Nobel Prize in chemistry for this technique in 1993. Now, we will go through the details of the PCR. First, we will go through the requirements of PCR. What actually the machine PCR requires. The first is the DNA template or the sample. And the DNA template is actually the double-stranded DNA that needs to be amplified. The second one is the primer, where small chemically synthesized oligonucleotides of about 10 to 18 nucleotides that are complementary to a region of the template DNA. And the third is an enzyme which will enhance the process of amplification and forming a polymer. Usually two common enzymes are used, that are Taq polymerase, which is isolated from thermophilic bacterium Thermus aquaticus, and Vent polymerase, which is isolated from Thermococcus litoralis. Okay, now these two enzymes are used because they can act at a very high temperature. They are capable of carrying out their activity at a very high temperature. Now if you go through the diagram, now we will go through the various steps of PCR. There are mainly three steps of PCR. The first one is the denaturation, where the double stranded DNA is denatured by subjecting it to its high temperature of 95 degrees Celsius for 15 seconds. This breaks the hydrogen bond between the two complementary strands of DNA and creates single stranded DNA. And both the single stranded DNA actually acts as a template DNA for the DNA synthesis. The second step is the annealing. The two set of primers are added which actually anneal to the 3' end of each separated strand. And what are primers? Primers are short sequences of oligonucleotides. These are added, which are actually complementary to the template DNA. Okay, and these primers also initiate the replication process for amplification. The third step is the extension, where DNA polymerase extends the primers by adding nucleotides complementary to the template provided in the reaction. Now, a thermostable DNA polymerase, which is the Taq polymerase, is used in the reaction, which can actually tolerate high temperature of the reaction. And these steps are repeated many times to obtain the several copies of the desired DNA. Now, this PCR is a very useful process. Since the wherever the amount of the sample is too less, we can use the PCR to carry out various number of cycles to get the amount of the sample more. Okay. So, this is important. Now, next we will come to the next step, that is ligation of the DNA. Ligation of the DNA. Next we will come to the ligation of the DNA fragment into a vector. Now, the vector DNA and the source DNA are actually cut with the same endonuclease to obtain the sticky ends. These are then ligated by mixing the vector DNA, the gene of interest, and the enzyme DNA ligase to form a recombinant DNA. The next step is the insertion of the recombinant DNA into the host cell organism. And this is done by the introduction of the ligated DNA into the recipient cells, which occurs by the several methods, before which the recipient cells are made competent to receive the cell. That means the recipient cell has to be competent. That competent here means that it has the ability to accept the foreign DNA. Okay, if the recombinant DNA carrying antibiotic resistance, like suppose ampicillin, is transferred into the E. coli cells, the host cell is transferred into the ampicillin resistant cells. Now, the ampicillin resistant gene in this case is called a selectable marker. Now, on growing transformed cells on agar plates containing ampicillin, only transformants will grow and the others will die. Next is the culturing of the host cell, and these are grown in appropriate nutrient medium at optimal conditions. The DNA gets multiplied and expresses itself to form the desired product. The next step is the extraction of the desired gene product, where when a protein encoding a gene is expressed in a heterologous host, which is called the recombinant protein. Now, the cells having genes of interest can be grown on a small scale or on a large scale. If it is grown on a small scale, the cells are grown on cultures in the laboratory and then expressed as a protein, extracted and purified by different separation techniques. But if it has to be grown on a large scale, the cells are grown in a continuous culture system in which fresh medium is added one side to maintain the cells in exponential growth. If you remember that there are various phases of growth of the bacterial cell. So, the lag phase, the log phase and the stagnant phase. So, exponential phase is the log phase, where the with exponential growth of phase of the bacterial cells is maintained and the desired protein is collected from the other side. That means it is a continuous process. It is not a stagnant process. From one side it is being added and from other side it is being extracted. So in large scale method, larger biomass is produced, which leads to high yield. The last step is the downstream processing, where all the processes to which a product is subjected to before being marketed as a finished product. This particular process is known as downstream processing. And this includes separation of the product from the reactor, purification of the product, formulation of the product with suitable preservatives, and quality control testing and clinical trials in case of drugs. Now, downstream processing is actually done by various bioreactors. Today, up to this, we'll continue with the rest part of the chapter in my next videos. Thank you.